Hello students, in this video lecture we are going to study about DNA replication, how DNA replicates and how all those enzymes comes together and create two DNA from one DNA. For that, we'll be starting the lecture. In this lecture, we'll be studying about how things happens during DNA replication, how it starts, how it elongates, and how it terminates. So for that, first we would be seeing that every replication starts at a point which is called as origin of replication. At this point, the denaturation occurs and bidirectional DNA replication bubble creates. And the point is also called as ORI-C. And after that, enzymes like gyrase, initiator proteins, DNA helicase, DNA primase, they come together and they create a complex called as replication fork. In this diagram, we can see there are various steps of creation of replication bubble. In the first diagram, we can see that initiator proteins bind to replication origin. This point is replication origin sequence. And after this, the second step, initiator proteins would come and they'll start melting DNA. And after the second step, DNA helicase comes and DNA helicase comes and attached to the complex. And prior to this, in this initiator proteins, we would be having DNA gyrase also, the class of enzyme topoisomerase. And after that, DNA helicase comes, loads onto DNA, and then DNA helicase denatures the helix and bind with the DNA primer to form primosome. And after that, the primosome DNA primase comes and create small RNA primer, and the entire assembly is done. And this portion is called as replication bubble. And in this diagram, you can better see the half of replication bubble. This is replication fork, where we are having various things. Like we would be having two strands. One is lagging and the second one is leading. Leading strand has continuous replication throughout. On the other hand, when we talk in terms of lagging strand would be having discontinuous DNA replication, and it may see the formation of Okazaki fragments. And what Okazaki fragments are, later on we will be seeing. And after this, this is the model for the events occurring around a single replication fork of E. coli chromosome. Like in this first step, we are having a replication fork here and lagging strand and leading strand. And the fork movement is always from 5 is to 3 prime, due to which the leading strand is continuous in nature, as we can see here. On the other hand, the lagging strand is not continuous. It is the creation of DNA on lagging strand is in patches. Here we can see, like this is our DNA RNA primer, and this is a small stretch of DNA on the lagging strand. And after that, the second Okazaki fragment creation is happening. Okazaki fragments are small patches of DNA which are made on lagging strand. It was discovered by Rizzi Okazaki, due to which these fragments are known as Okazaki fragments. Although these fragments later on will go and will compile, later on they would go and they'll make our intact DNA with the help of DNA ligase. So in this diagram, we can easily see DNA is opened up. We're having DNA helicase here. We're having DNA polymerase, and the synthesis of DNA is taking place with the help of DNA polymerase on both the strand, that is leading and lagging strand. In the second diagram, we can see it is further replication process is going on, and this helicase is further opening the DNA strand. Like leading strand, as we have already spoken, it would go continuous in nature, as we can see here also. On the other hand, the lagging strand is still making its DNA in patches. Like first, the Okazaki fragment is there, second Okazaki fragment is there, and formation of third Okazaki fragment is taking place. And in Okazaki fragment, there would be more than one RNA primer, because there are various Okazaki fragments would be made onto the lagging strand. On the other hand, when we see the leading strand, it would be having only one RNA primer, and the direction of replication is always 5:3. In the third diagram, we can see here Okazaki fragment formation is happening, and DNA polymerase is continuously dissociating, and then later on the process is still going on, and after third Okazaki fragment, fourth Okazaki fragment formation would take place. And after this, the fourth step would be DNA polymerase 1. As in the previous video, we had seen the functions of our DNA polymerase also. DNA polymerase 1, it replaces the RNA primer. RNA primer is of RNA nature, so it is mandatory to remove those RNA primers. For that, we are having DNA polymerase 1. So it would remove those RNA primer and make small DNA that is required onto that patch, and but still there would be some single strand gap left even after the functionality of DNA polymerase 1. And here we can see fourth Okazaki fragment formation is happening, and simultaneously DNA polymerase 1 is removing RNA primer from these strands. And in the last step, we'll find that RNA primer is being replaced with DNA polymerase 1, as DNA polymerase 1 is having 5:3 polymerization activity. So that polymerization activity would come into the picture, and it would make small stretches of DNA, which is left by the removal of RNA primer by DNA polymerase 1. And after that, fifth Okazaki formation would be there, and these gaps, which would be left even after the functionality of DNA polymerase, would be sealed by DNA ligase. And that is how we would be having two strands from one DNA duplex. And that is how DNA replication goes on. So to explain more into these things, we would be seeing two different mechanism of DNA replication. One is rolling circle replication. It is somewhat a different replication mechanism that is found in E. coli during the conjugation, and it is a mechanism which is prominently used by viruses for their reproduction. So in this diagram, in this rolling circle replication, we'll be seeing it that this is a circular DNA, and in the circular DNA, in one DNA, we get one cut, due to which we are having two openings, that is 5' phosphate and 3' OH chain. And in this rolling circle mechanism, we'll find one very peculiar feature, that it does not require RNA primer, because after the nick it is already having 3' OH end. So once it gets this 3' OH end, DNA polymerase comes into the picture, and it starts the synthesis of new DNA onto the circular DNA. And as we are seeing that new DNA formation is taking place, the old DNA is getting displaced. And this process would go on, and new DNA formation would take place. And this cycle may be repeated many times. And after the point when the new DNA reaches to the 5' end, the already pre-existing DNA would go, and there would be one cut here, and after which this the pre-existing DNA would go, and it would make a circular DNA, so which would be later on converted into double-stranded circular DNA. This process we can see, this process is very fast, as it does not require anything. It does not require RNA primer. It does not require so many enzymes. So this process is very, very fast, which is used by viruses for their multiplication. And these single-stranded DNA are known as concatamers, which are which later on which will go and which would make circular DNA. So this is one model of DNA replication, that is rolling circle model of replication, which is prominently used by viruses. And after that, we have one more model, that is Theta model, which was given by John Keynes. It was given by him, and he has called this model as Theta model, because the structure of DNA which he had isolated from E. coli during replication showed the structure which resembles like theta, a Greek letter. And this model was also helpful in understanding the bidirectional DNA replication process. So if we see this diagram, we can easily get it. In this model, we are having this double-stranded DNA, and it is opening because this is the origin of replication, and this DNA is opening, and it is going into both the direction, and the synthesis would take part, and lagging strand would be made in patches, and leading strand would be made continuously. And at each fork, DNA synthesis of the leading strand proceeds continuously in the same direction as that of unwinding. And, what John Keynes has done, he had taken E. coli cells and he had isolated DNA from those cells and kept those DNA onto the radiographic plates. And after some time, he had got some distinct shapes on the basis of which he had called this model as theta model. In this diagrams, you can see these are radioactive plates, and the diagrams he has got were very peculiar. And if we see the above part of this picture, we can get the same thing what he had seen in his radiographic experiments. And in this diagram, we can see this is a double stranded DNA, and here we are having one origin of replication. Now, this DNA origin of replication would open up, it would create replication bubble, and it would create the form, and it would create a open ending here, and DNA would melt somewhat, and after that, the fork produced around the circle. Now we are having replication fork, and these forks would go on to make new DNA, and ultimately two circular DNA molecules are produced. That is how one daughter cell is converted into two daughter cell, and the cycle would go on. So these structures and these structures which John got are very much resemble. And this structure, if we see, it resembles like a theta structure, and this model was very prominently used until it is used and proclaimed to be a very good model. And this model, in some after some modification, this model proved to be the experimental proof for the bidirectional mode of replication. This model has shown here that the replication goes on bidirectionally in E. coli. And with this, we are done with the DNA replication process. Thank you, and keep on watching our videos. Thank you.